So INFRAFRONTIER is a research infrastructure which aims at providing unique scientific resources, services, and expertise to advance understanding and treatment of human disease using mammalian models, mainly mouse models. Why we are here? So INFRAFRONTIER uses mammalian models for unraveling gene functions, aiming at the general understanding of human health and disease, not only rare disease, but also rare disease, of course. And the animal models are invaluable for biomedical research, especially in the context of rare diseases, because they have low prevalence, and in some cases it's really difficult to get data from patients, and also when the mutation is known, you can easily generate a mouse model with the corresponding mutation. Currently, we have, or there are 1,700 or over 1,700 mutant mouse strains potentially relevant for rare disease research available from INFRAFRONTIER. I will talk about this also later. But back to INFRAFRONTIER, so it's an European research infrastructure for the generation, phenotyping, archiving, and distribution of mammalian models. And the consortium is formed by leading European research institutions, which are mapped here, the ones in Europe. So there are in total 29 partners in 15 different countries, the ones you can see in this Europe map. And also we have partners in Canada and Israel. But we are also part of a global network, which is IMPC. IMPC stands for International Mouse Phenotyping Consortium, and it aims at the systemic phenotyping of knockout mice for all the around 20,000 protein coding genes in the mouse genome, which will generate a comprehensive catalog of gene functions. IMPC is making this phenotyping data and mice openly available to the biomedical community, and INFRAFRONTIER is the largest distributor of IMPC lines worldwide. We make around 300 shipments for IMPC strains each year, also worldwide. This systemic phenotyping is performed in the mouse clinics. Sorry, I forgot to mention here something important. So all these orange points are in Europe, are the IMPC European nodes, and all of them are also INFRAFRONTIER partners. So as I was saying, this systemic phenotyping is performed in the mouse clinics. They follow a standardized pipeline that is agreed among all of them to increase reproducibility. And they offer a whole system screen covering more than 500 parameters in different disease areas and organ systems. And one of the big advantages of this is that they can obtain maximum information using a minimum amount of animals. I told you that some of the IMPC mutant mice are available through INFRAFRONTIER, and this is specifically through the European Mouse Mutant Archive, or EMA. I will mention this several times today. So EMA is this archive, and here in this scheme you can see how it would work. So, but focus here not on IMPC strains produced by this big consortium, but for scientists in any research institution which maybe has produced a mouse model for a rare disease and wants to make it available to the community. So in this case, you would be the provider. You can submit this strain for archiving with us at EMA. You need to provide both animal material, so the animals and the data, information about this strain, phenotyping, everything that's available and can be of interest for potential other users. And then this, if you will look at the animal orange line, so the line will be cryopreserved, and if someone else is interested in getting this line, we can distribute it, and the recipient will get the line. But there's also a data flow. So as a provider, you also provide, as I mentioned before, all relevant information for this strain. This undergoes a process of data curation, which I will explain later, and is made available, and you can see it in our webpage if you're looking for a mouse model, and you can request it. So we will focus now on the lower part, the data part. There's three main ways how mouse strains or information about mouse strains can come into the EMA database. So IMPC lines is large scale production, is kind of automated, but then there's you as a scientist could submit your strain by using the submission form at our webpage, which I will show you after that. If you have several strains, we also offer the possibility to the researchers to provide information in a spreadsheet, and we can insert information directly into the database. Which kind of information do we record? So currently we have information for more than 7,600 mutant strains that are available from EMA. They all get a stable and unique ID, the MID, and then we record genetic properties, phenotypic descriptions, and annotations, disease associations, so DO, orphanet, important for rare disease, characterization, genotyping protocols, breeding and animal welfare aspects, publications related to these strains, using these strains, and also information about ownership and distribution and shipping logistics. So this is the submission form I mentioned before. So I will perform a live demo later on at the end of the webinar, but this is just to give you an overview. So it's quite easy and user-friendly form that you need to fill in with a lot of explanations, which information is asked, and different steps. Different tabs. You can always save between after completing one tab and continue on a different time point. We try to get quality data, and with this I mean in some cases we offer drop-down menus where users just need to select an option to avoid typos or wrong vocabularies. We suggest identifiers and ontologies like MGI, PubMed IDs, disease ontology, mesh terms. So this is what we do to try to get good quality data from the scientist, from the submitter. But then, as I mentioned before, we also do a lot of curation work. So first of all, manual curation at EMA. This means we have data curators, which will make sure that each strain submitted at EMA is properly annotated. So they will look at the genetic and phenotypic data and will curate and annotate them according to the guidelines of the International Committee of Standardized Genetic Nomenclature for Mice. They will also identify from published reference or any provided information and annotate of all the genes, alleles, expressed transgenes, backgrounds for each strain with the corresponding standard IDs, MGAs, gene symbols, web links, if applicable. And also, in some cases, if it applies, they also annotate and present the model human diseases as reported in reference or provided data according to disease ontology classification, which is also adopted by MGI. So I mentioned now a couple of times MGI. So MGI is mouse genome informatics, and they also do a lot of curation work. So MGI supplies experimental literature-based annotation to all mouse gene products, and also electronic base annotation. And our EMA curators are in touch with the curators at MGI. And if we get a submission for an allele or a gene that is not registered at MGI, they will take care of registering it. I won't go into detail here, but apart from this manual curation that I mentioned in the last slide, so we also have in place several mechanisms to retrieve phenotype and disease annotation from different sources. So MGI again, IPC, disease ontology, and we also get the ORFA codes and some information from ORFANET via the ontology lookup service from EBI. And all this information is saved in the database and also displayed in the INFRAFRONTIER website. Okay, so with all this, or in these last slides, I've tried to show you and explain that EMA data is fair, so findable, accessible, interoperable, and reusable. So INFRAFRONTIER EMA database has been selected as a verified data resource by the International Fair Sharing Consortium. And we also collaborate with the other European life science research infrastructures in the EOSCLIDE project. And there I'm at implementing and disseminating open fair data resources for biological and medical research in the EOSC, so European Open Science Cloud. So, and now I'm coming to EJPRD. So INFRAFRONTIER as a partner in EJPRD has set up during 2019 a rare disease landing page, which I will show you after that. This is a page where rare disease researchers can find INFRAFRONTIER information relevant for them. For example, rare disease conference where INFRAFRONTIER is involved. And we have also implemented a rare disease resource. And there you can find EMA strains that are potentially related to rare diseases and publications about these strains. The INFRAFRONTIER rare disease relevant information will be also accessible through the EJPRD virtual platform when this is in place. And we are also working in linking INFRAFRONTIER with other EJPRD resources, for example, GPAP. And in this sense, we had several productive discussions with our colleagues in Barcelona and Leicester about use cases that could also be useful for other EJPRD partners. This is the INFRAFRONTIER rare disease landing page. So you can find links to the INFRAFRONTIER rare disease resources here. So at the top, you have this link that will bring you to the list of mouse strains that are potentially related to rare disease, which lists currently 700 or more than 700 mouse strains with mutations in over 1,000 genes. And this has been linked or mapped to over 1,300 rare diseases. You can also find a link to the publications, the list of publications using all these strains, and also the INFRAFRONTIER and Rare Disease Conference here, which I mentioned before. I mentioned already a couple of times strains potentially relevant for rare disease research. What I mean here, or how we do this relationship, it's what I will explain in this slide. So we have our EMA strains, and they have MGI annotation, so with a gene ID, which gene is mutated in this strain. So we take the mouse symbol, and using ensemble, we map it to the human ortholog. And we use this human ortholog to query via OLS the ORFA codes, and then we do this way with the mapping of EMA strains to rare diseases. This information is now displayed in 2 different locations in the INFRAFRONTIER webpage, so in the rare disease resource, but we also highlight this information in the central EMA strain search. I will show you both later. And this will also be available in the future in the EJPRD virtual platform, and hopefully through or via other EJPRD resources. Yeah, I've talked now about a lot about data now. So I will come now to the live demo. So let's, and we will focus first on this part here. So rare disease researcher potentially interested in a EMA strain, how you could order it and where you can find information. So this is how I phrase it here. So looking for a mouse model for my rare disease of interest and placing an order online. So I will jump now to, I need to, okay, I need to exit full screen. Now here, can you see my browser now?
Yes.
Okay, thank you. So this is the INFRAFRONTIER main page. So we have links to different, or access to different services here, but one of the things we have in our main page is the central EMA strain search. This is not the rare disease resource. I will show you next step, but now we will also look for a rare disease here. So as an example, I've selected this rare disease. Oh, no, it should be 334. Now 10 strains. We have 10 strains potentially related to this rare disease, which is, okay, you cannot see it here, so, but if we select one of the strains, like for example, this one, this is a community strain. We call community strains the ones coming from individual scientists, not from IMPC, from large consortium. And you can find here, so if you look at one of these strains, you can find general information, so MID, gene that is mutated, name, which material is available from EMA, so frozen embryos and derivation of live mice in this case, information about the provider, who is the strain producer and owner, which type of strain it is, genetic description, phenotyping description, papers using this mutant, information from EMA. This is the EMA node because EMA is a distributed repository. So we have several archiving centers spread across Europe. And this strain is archived in Stockholm. And in the papers, but if we go to the last part here, we have the disease, phenotype and disease information, and we can find here this ORFA code that I looked for, I typed. This is familial atrial fibrillation. So it's just a random rare disease I selected for the demo. But you can also find now in the central EMA strain search, not rare disease specific, these ORFA codes and the rare diseases here. Now I will go to the landing page. I've shown you before a screenshot, so you can find it if you go to our main web page, knowledge-based therapeutic area and rare disease, INFRAFRONTIER rare disease. This is where it's located, but so I have it here, and you have the different sections here. If you go, the first one is this strain. Okay, I already looked for it before, so if you go to this the place where you can find the strains related to different rare diseases, you can also type here the ORFA code. Okay, and it's also found, and we can see the same 10 strains that were found before, with a short description about the rare disease from ORFANET, the genes that are mutated in these 10 strains, and the reference related to these 10 strains. I've shown you before, so maybe I take the same strain that I've shown you before. So, and you will see it brings you to where we were before, and it's displaying exactly the same information in the same place. But I want to show you now a different type of a strain, an IMPC strain. This is coming from this big consortium doing systematic phenotyping. In this case, if you go to information from provider, under genetic, in this section, genetic description and phenotypic description, you have links to IMPC portal for phenotyping description, for example, if we follow this link, you will be directed to the INPC portal, and you can see the phenotyping that has been performed to this line. You have this nice overview here, which systems have been tested. So gray not tested, in blue not affected, in red significantly affected. But you can also then see a summary of the phenotypes, the significant phenotypes here, but you can then download in different formats. It's loading. You can see, usually it's quite fast. You can see all the data from this phenotyping, and you have access to it and can download it. It's strange. I don't know if it's my connection, but usually it loads quite fast. Okay, so it was. I will go back to our web page. So just to show you that this way you can have for the INPC lines, you can have all information available from INPC, and it's directly linked in our strain search. Let's see how to order it. Let's imagine you're interested in this strain because it has a mutation in the gene that it's related or it's affected in the rare disease you are studying, and you look at the INPC portal and you see the mice have a similar phenotype as your patient, so you would like to order it now. So you just need to fill in this information, recipient information, shipping contact, and billing contact. And then this is already pre-filled with the MID and the strain name. Then you need to select which material you want to get, frozen embryos or live mice, which type of user you are, so academic, commercial user. So I will explain you about this at the end of my presentation. Usually EMA distributes only to There's a third option, commercial service provider. It's a company.
I think we lost most 4 months for a few minutes. Thank you.
Can you hear me?
Yes, now we can hear you. We lost you for a few minutes after the academic display, and you were about to go to the commercial company. We will cut this part in the processing of the video. So you stopped at the academic user, and you were about to go to the commercial.
Okay, good. I was not talking a lot for a long time. So I just said that. Do you see my screen now? No. Yes.
Yes, we see it.
Okay. So I was explaining that I will explain later at the end of the presentation how commercial users may get access to strains in some cases, although usually EMA only distributes to non-profit users. And this third option is just in case a scientist uses a service provider to import and breed the mice for them, but they are not doing research. So we also have this. Important here, you need to check that you have read and understood the EMA repository conditions and the data privacy statement, and then you can send it. I will jump now to my PowerPoint again and show you what happens when you press the order button or the send button. So you automatically get an email, confirmation email with important information, and which I will summarize here. So what was written in the email, it's mainly a detailed quality control level applied to this strain because it differs. Although you can also see this before ordering, of course, but this is just to make it clear again, the confirmation email. A request summary is also attached, and if an MTA is needed, it will also be attached to this email. You are also told that the distributing EMA node will contact you soon, and the EMA node, our partners, when they contact you, they will help to organize the paperwork, MTA, permissions, courier for the shipment, and will clarify all the details for the request. So I just want to make it clear here, this is not just ordering a book at Amazon. So it involves shipping of live mice or cryopreserved samples, and so it's not a trivial process. So we've seen now how to order a line and look for your potential strain of interest and order it. But what if you are at the other side? So you are a rare disease scientist, have developed a mouse model, which turned out to be a good model for rare disease, and you want to make it available to the rest of the world. So you can submit it to EMA. And how this works. So this is what I've written here. So I have a model and I want to deposit at EMA. So now we go again to I don't know where I have this, but you can go to the main page again, and you have here this deposit mice. If you go here, you have you can go to this page where you will find all information or relevant information regarding depositing strains into EMA, and you also have a link to the submission form. And the first page you will find is this with relevant information about the process and this and things you need to know before submitting. Important here is that the submitter should be the strain owner, or at least have permission from all owners to deposit the strain at EMA. Only one strain per submission. It explains that in most cases sperm will be cryopreserved, but if needed, embryos can also be cryopreserved, and some other technical things. And then again, you need to accept EMA repository conditions, data privacy statement, and you can also subscribe for the newsletter. Then you need to enter an email. I will enter my email here and submit, start submission, and you are now in the submission form. So at this point, you get an email. And this means you don't need to complete the submission at once. You can just save it and go back to it by using this link. So this is the email you get when you enter your email and start the submission. You can use this link at any time to retrieve the submitted information and continue with your submission. And now back to the submission form. So it contains several explanations. So some of them here, in some other cases, you will get this information will come when you have your mouse over the field. As I mentioned before, we also have drop-down menus, for example, here for the background, so that you don't need to type it and maybe make some mistakes, so you can just select it from if not, you can of course specify it. In each of these tabs, you can say save and continue, and it will be saved. Maybe I can just type something here and show you how it looks. So there's some fields that are mandatory. They are marked with these red asterisks. Mutation induced. Okay, if I say now save and continue, this red cross turns into a blue tick. This means this step is completed. I can go now to next one. You don't need to do it in this order. You can now complete the third one and then the last one and then go back to the previous ones. At the end, you have this preview and submit tab where you can always export to PDF. In this case, you will get a PDF. It has already opened. So it's clear here submission is not completed. You will get here the whole submission form, the completed parts and the not yet completed parts. And this is quite useful because you can, if you are in the middle, you can just download it and take it with you, complete what's missing, check what we're asking for, ask your colleagues, and then go back to the submission form with the link provided in the initial email and submit the strain. Whenever you are done with all steps, you just need to press. I cannot do it now because I did not go through all tabs and fill the information. But if you have blue tick in all of them, then it's possible to press submit, and you will get again a confirmation email. And I go back again to my PowerPoint. So this is the information, the email, automatic email you get upon submission. And it mainly says that the information will be evaluated, but maybe I can explain it with this slide. So what happens upon submission? So first of all, before evaluation, we will make sure that we have all information that's needed. Nothing is missing, everything makes sense. If that's not the case, we'll contact you and ask for the missing information. If we have everything needed, then we will send the submission to the external evaluation committee. And this is the list of criteria or questions they get in order to evaluate each and every submission. So in parallel, we ask one of our partners, one of our archiving nodes, to check the technical feasibility of archiving the strain. And they have different criteria for deciding on this: health status, if the strain is clean or not, and so phenotyping, breeding issues that the strain may have, and if the strain is accepted by the external evaluation committee, then the user is informed, and the archiving node will take over and will organize with you all required paperwork. Again, this implies shipping live mice around or cryopreserved material in some cases. So it's not that trivial, but they are experts in this and they help you. And then when they get the mice, they will start cryopreservation. Once the cryopreservation process is started, the strain is visible in the EMA public strain list, but only for registration of interest. So it's not, you cannot order it, you can only register your interest. And this, the reason why we do this is to, in case there's some excess of mice during the cryopreservation process, they could be sent even if the strain is not completely archived yet. And also, at the end of the cryopreservation process, it would save deriving mice from frozen material. If we already know some scientists are interested in getting this line, they could get them right away. So, and a strain is considered fully archived, only when different quality control steps are done, and then we change this, and it's not possible to register your interest, but you can order the strain directly. At this step, when a strain is fully archived, so all users that registered the interest in the past, they will get a notification so that they can turn their registration of interest into a real order. Yeah, I mentioned both when ordering from EMA and when submitting a strain to EMA, the EMA repository conditions. So this, the submission distribution of mouse mutants is subject to these legally binding conditions. And it's important for strain submitters to know that the ownership stays with them. EMA lines, as I said before, they are supplied to qualified researchers as a service to the scientific community solely solely for non-profit research purposes and not for commercial use. Here, there's some collections that can be made available through the to commercial users via an external company, and also for communities strains. If we get a request for a strain from a company, we'll always put in contact the company with the strain owner. If they get a licensing agreement, then we will service this request. If not, then we won't, of course. Recipient, it's also important and written in these conditions that recipients may not transfer or sell these mice or their progeny or modifications, and any existing MTAs will remain in full force and effect. And you can also see the MTA documents and download them from our webpage and in this link. So since the slides will be available in this link, you can see the EMA conditions. And coming to an end, I've talked at the beginning about this systemic phenotyping. I've talked a lot about the EMA repository, but these are not the only services that INFRAFRONTIER can offer to researchers and rare disease researchers. But we also have partners specialized with a lot of experience in model development. As I said, if you know exactly which mutation is causing the rare disease you are doing research on, and there's no model available, they can generate it for you. We're also offering transnational access calls, training and consulting, and therapeutic areas. I've also shown this because one of these areas is rare disease. I will show these therapeutic areas, the ones we are focusing on, in the next slide, but I want to mention here what it's important that it's a distributed research infrastructure, but everything is available through this INFRAFRONTIER webpage, and we show one face to the customer, although we are offering so many different services, and we have so many partners. Next slide. Okay, no. I mentioned we have special resources and landing page for different therapeutic areas. One of them is rare diseases, which I've already shown you, but we have also responded last year to the new pandemic and have now a list of several resources and partners that can help and support COVID-19 research. And we also have a lot of resources for cancer research. And with that, I'm at the end of my presentation. Thank you very much for attending, and please do not hesitate to contact us if you have any question, if anything was not clear and you prefer asking us by email, or if you want to use our services, so please contact us.